Alrighty, let's start with chapter 1, the science of biology. So, by definition, biology is the scientific study of life. So there's some characteristics, there's some characteristics that if an organism has all of, it's considered to be alive. So if the organism has some form of order, also has the capacity to reproduce, has the capacity to grow and develop, process energy, regulate themselves, respond to their environment, and the potential for evolutionary adaptation, it's considered to be alive. So all living organisms have all of these characteristics. So an example of evolutionary adaptation is one demonstrated by beetles and birds. So let's say you've got an area containing a population of beetles with varied inherited traits, and these inherited traits dictate their color. So we have some darker beetles, some medium colored beetles, and some lighter beetles. And they're all living in this hypothetical black backdrop of a space. Now, the lighter beetles have a higher contrast with the black backdrop, so they're probably easier seen by predators, and so most of them are going to be eliminated or eaten because of their traits. So they're birds are better able to see them, so they're eaten more frequently. And remember, genetics is what's determining their color. And so over time, birds mostly ate the lighter colored beetles, and now the majority of the population is all dark. So that's a simple example demonstrating evolutionary adaptation. So here's a summary to kind of discuss what all of these terms mean, or what these things mean. So order, what is order? If it has some form of organization, you could flip that with organization. Cells are typically organized in specific ways if they have the potential for reproduction. So the ability of organisms to reproduce their own kind. So that could be through sexual reproduction or asexual reproduction, just a cell splitting in half to make two cells. Growth and development. So cells are a great example. They grow, they'll shrink, a larger multicellular organism will grow and develop. And this is all dictated by gene expression, or DNA. And then energy processing, so the use of chemical energy to power the organism's activities and chemical reactions. And then the capacity to interact with the environment, so response to the environment. So you could regulate your body temperature, you could take things into your cells, you can throw things out of your cells. And then regulation, so the ability to control an organism's internal environment with the limits that sustain life. And then we have evolutionary adaptation, which I already went over. Okay, so these are all the things that constitute what is living. So if an organism or a thing has all of these characteristics, we generally think of it as living. Now let's talk about the organization of life. So we're going to start with not the very smallest unit of matter, but we're going to start with small, and then we're going to move up into the way we organize living things. So we can start off with atoms. So not the smallest, but because we still have subatomic particles, but atoms are pretty small. Atoms congregate together, form molecules. Those molecules will eventually, they can group together, and we can form these things called macromolecules, which we're going to get into in chapter 3. So macromolecules, macro meaning large, molecules, so just large molecules. An example of it is DNA, which is nucleic acid. Now we have many macromolecules kind of working to do the same thing, not working necessarily, but functioning to do the same thing. And we get these things called organelles. So these are kind of like the organs they're synonymous with the organs of your body, kind of parallel to the organs of your body. They're the organs of the cell. So, your cell has many different parts, a nucleus being one of them. So your nucleus is considered to be an organelle. Now, conglomerate of many organelles forms a cell. So you have a nucleus, and you have ribosomes, and you have endoplasmic reticulum, and you have your cell membrane. Well, now you've got a full cell. So you move up to cell. Now, cells, a bunch of cells working together, kind of conducting the same thing, that's when we get a tissue. So nervous tissue is a great example of this. Many neurons working together to conduct kind of the same thing, the same process, the same activity. And then you have a bunch of tissues that work together, or that form an organ. So the brain. It's a collection of brain tissue, which is a collection of neurons. And then you have organ systems. So not just the brain, but you have your brain and your spinal cord and all of your peripheral nerves throughout your body. We just call that whole thing the nervous system. And eventually we get to the organism. So it's a collection of organ systems. Now we've got one organism. Now if we take a bunch of organisms of the same type and group them together in a space, or just group them together, we call that a population. So a bunch of elephants grouped together in a space, it's a population of elephants. Now if we're talking about elephants and ostriches in a space, we have two different types of organisms, two different species, we call that a community. And then as we get larger, or actually not larger in space, but once we start to include the abiotic factors. So your elephants are biotic, they're living. Your ostriches, biotic, living. Now once we're talking about temperature and elevation and rainfall, now we're talking about an ecosystem. So an ecosystem is not just focused on the living organisms, but the living organisms as well as the other factors playing a role in that environment. So that is temperature and elevation and rainfall. Oh, and then lastly, we have the biosphere. So that's just Earth, the biosphere. So this is a summary to, you can look at this later. This is just to help you guys recap everything. Okay, so ecosystems. In most ecosystems, plants are the producers. So towards the end of the quarter, we're going to get into ecosystems. When I mention producer, I mean something that photosynthesizes, something that will produce its own energy that photosynthesizes. So I guess the most common example we could use are plants. Plants are producers. Plants are the producers. There are photosynthetic bacteria. We usually can consider those producers as well. So they provide the food for an ecosystem. Then you have consumers. So these are things that eat producers, or things that eat other consumers. So consumers eat plants and other animals. And then we have decomposers. Those act as recyclers. So that could be bacteria, that could be fungi. So these break down the matter of dead things into their base components, which are like molecules and atoms. They'll break them down into simple mineral constituents or nutrients. Okay, so ecosystems, this is just kind of a overview. Okay, so we have producers, plants, and those producers are eaten by consumers. So you have your giraffes that'll eat the plants, and then you have, we could just say carnivores in general would, eat the giraffes or whatever. Those are consumers. So in the process of eating the plants, they're receiving energy. So these arrows denote energy transfer. They're receiving energy, but in the process of that, which we'll get to in the second section, they're losing some to heat. So to process any kind of food that you eat, you're also producing heat simultaneously. So they produce some heat. And, the carnivores eat the herbivores. They produce some heat. in the breakdown of those carnivores or the breakdown of the dead bodies of the herbivores, heat is produced. And so your decomposers break down that stuff, that material, into its base components. So abiotic chemicals, so things like carbon dioxide and oxygen and nitrogen and other minerals, which are then taken up by plants and assimilated by the plants. All of this yields heat. So keep that in mind as we get further into the quarter. Oh, so what I've left in here are questions for you guys to answer if you were in class or if we had a synchronous. Okay, so I'll just read it and answer it for you guys. This is for you guys anyway. So, life's organized in a hierarchical fashion. Which of the following sequences correctly lists the hierarchy from least inclusive to most inclusive? So this is the organization of life. Remember, we covered two things earlier: what it means to be alive, what characteristics you have to be alive, and we also covered how are living things organized. So let's go from least inclusive, so the smallest, to the largest or most inclusive. So A, we could eliminate that right off the bat because it says ecosystem. We know that that's the most inclusive, so it's probably going some inverse way. We got cell and then molecule. That's another red flag. You go from more inclusive to less inclusive, so that's wrong. Here we go. We got molecule, organelle, cell, tissue, organ system, organism, population, community, ecosystem. So C would be the correct answer. So, we're missing atom, but it still moves from the least inclusive to the most inclusive, given what's in the list. Okay, so I would have made it to C if I was taking a test. But I would have probably still read D, so what does D say? Organism, organ system. Okay, so this is a pretty easy test because these are just blatantly wrong. No tricks there. All right, so how scientists think. All right. Scientists. The word science is derived from the Latin verb meaning to know. We're talking about the different forms of reasoning. So, we're gonna talk about two different forms of reasoning here. So scientists will use inductive reasoning to draw general conclusions from many observations. So this type of reasoning moves from specific to general, meaning you within your life and your experience are experiencing many specific things, and you use that data of those very specific experiences to apply to the universe. Universe, very general. Or you can even just apply it to the biosphere if you wanted to be less general. So naturally, because you're using experience, your personal experience is inherently uncertain. Now, deductive reasoning, this is kind of the inverse. So deductive reasoning is used to come up with ways to test a hypothesis, and a hypothesis is a proposed explanation for a set of observations. But to break down deductive reasoning, what you're doing is you're taking things that are known about the universe, and you apply it to something specific. And I think I might have an explanation for this in the next slide. Okay. So classically, if you were to take like an intro to philosophy class, the way they break down inductive reasoning is all of the swans we have seen are white. So in my lifetime, all of the swans that I've ever seen are white. So all of my specific experiences generate this conclusion that all of the swans I've seen are white, or actually that is my experience. Therefore, I can conclude that I expect all swans to be white in the universe. So I'm taking specific experiences and I'm applying it broadly to the world. Therefore, I expect all swans to be white. Now, if we're using deductive reasoning, it's a little bit more precise. So all humans are mortal, and this is kind of derived from Philosophy 101. All humans are mortal. So this is your first premise. Donald Trump is a human. That's your second premise. Therefore, Donald Trump is mortal. So all humans are mortal. That's big picture. Now you're applying this or no, and then Donald Trump is a human. That's another big picture thing. That is something that is a consensus. That is something that we know. And then you apply it to something very specific. Therefore, Donald Trump is mortal. So the conclusion is true if the premises are true. So deductive reasoning is a little more precise than inductive reasoning. Okay, so scientific method. So here I'm going to give you guys an example of an experiment that was done to kind of walk you through how the scientific process happens. So an observation, or it is observed that the amphibian populations are rapidly declining. So amphibians dying, cool. So the next step is, so you got your observation. Next step, you ask a question, do some background research. So here's some questions. What's causing the decline? Maybe, is there a fungal disease that's present? Maybe higher ultraviolet radiation is causing decline. Maybe it's an agricultural chemical. So these are just some questions, some postulations. So after that, we create this hypothesis. So a hypothesis is a testable explanation, not a question. It is an explanation. So a study that was done using this data, so there was an actual study done by Tyrone Hayes at UC Berkeley. He noticed amphibians population was declining, and his hypothesis was exposure to atrazine during larval development causes abnormalities in the reproductive system of frogs. So that's a testable explanation. So coming up with a hypothesis could be pretty easy. It's coming up with a way to test it that's a little bit more difficult. So his prediction was that if we expose larval frogs to atrazine, so larval meaning baby frogs to atrazine, they would develop reproductive abnormalities. If you got reproductive abnormalities, then probably you're not going to have success reproducing. Therefore, that's probably a pretty good explanation for why the amphibians or frogs, the population of frogs is declining. So this is how he tested his hypothesis through an experiment. So we had 3 tanks. All of them had different conditions. So remember, atrazine is what he was testing. So you have 3 tank conditions with all conditions constant. So temperature, volume, the water is probably the same, except atrazine concentrations. So water is the same, so meaning that there's no other chemicals, but what he did was he added 0 parts per billion to one of the tanks, 0.1 parts per billion to another tank, and 0.25 parts per billion to a third tank. So the 0 parts per billion, if any of you guys know what a control is, the 0 parts per billion is used as a control group. So you have 3 different tanks. You add atrazine to 2 of them. That's your experiment. Those are your experimental groups. But you need a reference point for adding nothing. So you need normal frogs to be able to compare. Let's just say you're expecting for them to have some weird tumors in their genitals. You need frogs without tumors in their genitals to compare the tumor genitals to. So that's the purpose of your control group. Every experiment needs a control group. So this is your control, and these are your experimental groups. So they placed 10 tadpoles in each tank, 10 tadpoles in here, 10 tadpoles in here, 10 tadpoles in here. And once they got to their adult stage, they sacrificed them or killed them, and then looked at their reproductive tissues. And so what they found was that there was a correlation of the degree of atrazine exposure with the presence of abnormalities. So that means that the more atrazine that they were exposed to, the higher incidence rate of reproductive abnormalities. Oh, I didn't even give an explanation for this. So these are the testes. So within the testes of the frogs, eggs developed. That's not really conducive to reproductive success. So it really mostly affected the male reproductive system. Okay, so conclusions. So lastly, we have conclusions. Accept or reject hypothesis. So once you come to your conclusion, within that conclusion, you must accept or reject your hypothesis. Those are the steps that must be taken after that is the thing that you are doing within your conclusion. Do you accept or reject your hypothesis? In this case, they accept their hypothesis. Still a successful experiment, even if you reject your hypothesis. So let's say atrazine didn't cause any reproductive abnormalities in the frogs. It's still a successful experiment because you learned that atrazine doesn't cause reproductive abnormalities. So exposure to atrazine at concentrations as low as 0.1 parts per billion induces abnormalities in the male reproductive system of frogs. So here's a little chart to kind of overview that. So you have your observation, amphibian population declining. Ask a question: is it something in the environment? Do background research. Look up all of the potential things that could be within the environment. Oh, atrazine is listed. Construct a hypothesis. I think that atrazine could be causing this. Then you test it with an experiment. So you run that experiment, and if the procedure is working, and you analyze the data, you find that there's, in the testes of frogs, you're getting some, ovaries developing within the testes, then your results align with the hypothesis, so you can accept the hypothesis, and you communicate the results. So hypothesis is an educated explanation based on an observation that could be tested by experimentation and is falsifiable. So it can be proven incorrect, which is fine. A collection of related hypotheses that have been supported contribute to a theory. And so 4 major theories, and then I have 3 listed. So some major theories that we'll probably discuss in this class, is cell theory, gene theory, and the theory of evolution. So I should probably write 3 up there instead of 4. And then here's something that's important for you to know. I'm telling you now, this is going to be on the final. This is going to be in probably your first exam. Know the 3 domains of life. So the very last section will probably talk about the distinction between these 3 groups. But just to give you a head start on this, we classify living organisms into 3 domains. So we've got bacteria, which are bacteria. They're these single-celled organisms with no nuclei. And then we have what we know as eukarya. And so this is all the stuff that we're made of. We are eukaryotes, types of eukaryotes. Fungi are eukaryotes. Animals are eukaryotes. Plants are eukaryotes. Some single-celled organisms with nuclei are prokaryotes. And then we have archaea. So archaea, I would say that they're close in morphology to bacteria in that they're probably about as small. However, the main difference between bacteria and archaea is that archaea usually live in extreme environments. So extreme acidity, they live within extreme acidity or extreme temperature environments. And we'll probably talk more about that in a future lecture. So just to talk a little bit about the name of eukarya, eukarya can be translated to true seeded. And now I mentioned nuclei earlier when talking about bacteria and eukarya. Eukarya have nuclei. Bacteria do not have nuclei. Neither do archaea. So that is one thing to make a little bit more of a distinction. Okay, so we have a question. Yeah, final question. So a scientist performs a controlled experiment. What does this mean? So the experiment proceeds at a slow pace to guarantee that the scientist can carefully observe all reactions and process all experimental data. To be considered a controlled experiment doesn't necessarily mean anything about the pacing. You could do an experiment fast, you could do an experiment slow. It could still be a controlled experiment. So I would say that this is not a requirement in order to have to perform a controlled experiment. This is not a prerequisite for a controlled experiment. One experiment is performed, but the scientist controls the variables. One experiment is performed, but the scientist controls the variables. So one experiment is performed usually when you're running a controlled experiment. Your control group counts as a type of experiment. So this one's incorrect. One experiment is performed. So let's say you were to run an experiment using the example that we used earlier with the atrazine. Let's just say you had one bucket or one test tube where you threw a bunch of atrazine in and you threw the eggs or the tadpoles in there, and then you saw the results. You have no control group, so you wouldn't have anything to compare it to. So whether or not they developed reproductive abnormalities, you still have no proof that this isn't some kind of genetic deformation in the tadpoles that you were using. You need a control group. You need a mode of comparison. So this one's false. You want at least 2 experiments. Oh, here we go. So C, at least 2 experiments are conducted, one differing from the others by a single variable. Yeah. So the variable in this case would be atrazine. So you have a control group with 0 atrazine, so that the variable being atrazine, 0 atrazine, and then you have another group with more or less more atrazine. So 0.5 parts per million of atrazine, for example. Okay, so C would be the correct answer. Let's look at D though. So the experiment is repeated many times to ensure that the results are accurate. That's a good general rule of thumb. When you're running experiments, you want to run it many times, but it's not a requirement for a controlled experiment. What I'm doing here is asking you guys to define what a controlled experiment means. Or yeah, define controlled experiment. C is the correct answer. That's just a general good rule of thumb. Same with that. All right. So hopefully you learned something, and I'll see you next video.